Hello, everybody. Welcome. We'll get started at about 3 after the hour to allow others to sign on. Hello, everyone. Welcome. We'll get started at about 3 after the hour. Looks like the attendee count keeps going up, so we'll just give it a couple more minutes. We'll do just one more minute. Right, it's 3 after the hour, so I wanted to go ahead and get started. Welcome, everyone. I'm so pleased to see so many joining us today from around the world for the first Variant Scientist Network presentation. Today we'll be having a roundtable, but before we get started with that, I wanted to give a little background into the Variant Scientist Network and tell you a little bit about how we got started. In March of 2023 is when the Variant Scientist Network got started. It was started at the first Variant Scientist session that was presented at ACMG, where we launched the Variant Scientist Survey. In March of 2024, we had another Variant Scientist session at ACMG that included case presentations from a variety of variant scientists. And in July of 2025, we published our Variant Scientist Survey, which I wanted to just touch on briefly. This is the first figure of the Variant Scientist publication. It included demographics of the respondents to the survey, their ages, their education levels, and what they studied, as well as the work environment that they currently participate in. I think most importantly to many of us was the salary survey that showed the level of experience that the variant scientists have, as well as the salaries that they receive. And the next most important thing, I think, is just showing what variant scientists do in addition to investigating the clinical significance of variants that are detected by genomic sequencing. So after that survey was published, we had our first Variant Scientist Network Advisory Board meeting. Advisory Board members were solicited by an application process and were selected. And we had our first meeting in October of 2025 that helped establish the goals for the network. And one of the goals for the network was to have education and networking events. And so today we have our first networking event of the Variant Scientist Network. But what were the other goals that the advisory board decided that the Variant Scientist Network should focus on? These are what the advisory board decided should be the key priorities for the Variant Scientists Network. They include education and networking, which today is the start of. They also included a job board and a discussion board possibility. Those have both been undertaken in starting a LinkedIn site. If you're not yet part of the Variant Scientists Network LinkedIn and you'd like to be, I think you can search for it and find it that way. There's also a Slack channel that's been started for communication amongst variant scientists. Um, I'll share that link at the end so that you can be signed up if you want to receive our quarterly updates, or if you want to be part of the Slack group or the LinkedIn group. All that information will be provided to you. A mentoring program is still in development and hopefully will be launched in 2026. But for the agenda for today, we're going to have 3 variant scientists present, starting with Yasmin, who is a clinical specialist in molecular biology, and then we'll have Rachel Mercado, who is a genomic variant scientist, and Trisha Hebron, who's a variant scientist. And just from that, you can see that we all have very different titles, but we all have very similar kinds of daily work. Please use the Q&A box for any questions that you have during each individual's presentations or introductions, and at the end we'll have a Q&A session that I'll help moderate. So with that, I will hand it over to Yasmin, who will talk about her experience as a variant scientist.
Thank you, Alexa. So I'm really happy to see this great turnout today, and I'm really happy to be part of this first roundtable session on career trajectories. I was invited to speak as someone who, as a variant scientist, has a PhD. So I have a PhD in molecular biology, and I'm going to talk to you about how I got to where I am today as a clinical specialist. But also I want to talk about it from a perspective of someone whose trajectory has not always been a straight line. So my trajectory has really been filled with a lot of twists and turns, ups and downs, and pauses and reflection. And coming to you as someone who has a background in research of about 20 years. So I really did this huge, it's not a huge pivot, but I would say a pivot of going from research to variant scientist, and kind of like the steps I went through to get where I am today. So as I mentioned, I have a PhD. I'm in Canada. So I don't know how many of you are familiar with the French part of Canada. It's Quebec province in Canada that actually today our premier just stepped down. So it's a big day for us because healthcare is very important here. We work with a public system. So basically we, through our taxes, we pay through our taxes for our healthcare. And so generally, everyone equally is welcome to have healthcare, but that also means that I work in a public system where, I work in an environment which might be very different from where Trisha or Rachel, our 2 other panelists, work in. So I think that's something I will also talk about a little bit later on. Okay, so when I was doing my PhD, I absolutely loved it. It was where I wanted to be and needed to be at that point. I was learning all about, the basics of how to read papers and be discerning and look at Western blots. But I was also understanding things like, gene function and pathways and evaluating evidence. And so I think these are really important skills that I used all through my research career and my grad and my post-grad, but I also use it today as a variant scientist. So when I was nearing the end of my PhD, unfortunately, I had some health challenges. I had an accident. And so I couldn't pipette the way I used to. I couldn't stand at a bench the way I used to anymore. And it was really hard for me because I had to kind of let go of that identity as someone who was a researcher. And so I had to stop. I had to pause and I had to really reflect very deeply. What did I want and what were my needs? I needed something that, suited me physically, but also suited me mentally as well. So something that was challenging. And I wanted to absolutely to stay in science. So this is where I set out to speak to as many individuals as possible in the science realm. So I would say I must have talked to a dozen people, from law in the medical sciences to, so many people. And I just kept searching. And one day I came across a newspaper article talking about genetic counseling. And I fell in love and I became obsessed. I thought to myself, all right, I want to be a genetic counselor because I want to help patients. That was my number one, but I also wanted to keep my background in what I already knew. That was really important to me. So how could I use what I already knew in biomedical sciences and genetics, in molecular biology, to help patients? And this was the answer. So I set out to be accepted into a genetic counseling program. And it took 5 years and 3 tries. And finally, I got in. And that is when I was like, this is it. I found my path. I knew. My program was wonderful. We were a group of 5 in our cohort, and it was great. I spent a lot of my time counseling patients. So from the get-go, my rotations allowed me to sit with patients and understand their journeys, understand how, to interact with them, but understand also the clinical aspect behind what was happening. I listened. I learned to, have that empathy for patients. And that was, I would say, the bread and butter of the program. But also we were allotted time to be able to give presentations and, and research project. And I think that was when I really started to pick up on variant interpretation. And I think it probably comes from my background and my graduate work and research and all that, is that I saw variant scientists, variant interpretation as familiar, but also still exciting to me. It was like a puzzle. I started to give presentations during rounds about variant interpretation and how genetic counselors dealt with them in the clinic, and started to learn about VUSs, so variants of uncertain significance. And I started to understand, I said to myself, oh, look at all the studies that go behind interpreting a variant. I look at those papers, and with my background and my PhD, I'm able to quickly read and discern evidence that is, essential and important in interpreting those variants. So it became like a passion of mine. And I'll be honest, when I finally graduated with my master's in genetic counseling, I didn't know what I would be doing. I imagined I would be a genetic counselor. But in Quebec, it's very difficult to land a genetic counseling position. As I mentioned, I'm in the public system, and it's very difficult to get clinical positions. We are very restrained by limitations. We have a lot of limitations on our resources. And so luckily, I ended up being recruited by the clinical diagnostic laboratory. And they said to me, with your PhD and now you have your clinical experience, you would be a perfect fit. We are going to recruit you. We will train you on the job. What do you think? And I thought, this is great. And I think it was a very opportune time to take this position because I have a family. At home, I have a daughter and a husband, and sitting in an office and being able to do this work in a quiet space for me was also something that was a great need for me, that I saw as a great positive. So I said, let's do it. And here I am 2.5 years later. I'm still in this position. And I have to say that, there's a lot of challenges. I think we must do something like, I don't have the exact number, but I know it's in the tens of thousands of tests per year we do a lot, and we're a small team. So there are challenges, but at the same time, we have a great team. I think it's important, to appreciate your coworkers. I work with a team of a bioinformatician, molecular geneticists, lab technicians, and we all work great together. And I really appreciate the environment that I'm in. Um, right now, my focus is on somatic variant interpretation. So, we test the tumor tissue to look for variants, targeted variants and genes that will help us uh, diagnose or, give some sort of therapeutic indication or even prognostic information. I would say my most favorite part of my day-to-day is that I am building a targeted gene panel for inherited retinopathies. And so I think I've curated something like 500 genes. So gone through those genes, looked at the evidence, should they be on the panel or not? And, determining which genes are difficult to sequence and trying to work around that. And so I think that's also where my PhD comes in, is I understand some of the technical aspects behind, running these tests also. So that comes in very handy for me. And so, yeah, 2.5 years later, here I am. We're growing. We're always building new tests, which is also a challenge. And I've learned so much along the way, and looking forward to, continuing. So thank you for listening.
Thank you so much, Yasmin. That was fantastic. And it really highlights some of the paper's findings that we saw, that variant scientists have their hands in so much of the testing and are part of a collaborative group of multidisciplinary fields. I appreciate that perspective.
Great.
Next,
Thank
we'll
you.
have, thank you, thank you. Next, we'll have Rachel Mercado share with us.
Hey everyone. So I have a small presentation. Yeah, so my name is Rachel Mercado. I am currently a variant scientist at Quest Diagnostics, and of course I'm like speaking as a variant scientist, not like an employee of Quest at this moment. Just as a disclaimer, and I want to discuss kind of my journey to variant science. Um, like Yasmin, I kind of didn't have a straightforward path as well. It wasn't like I knew this was a thing and got into it. So my education background is that, well, first of all, I've always loved science. I always knew I wanted to be in healthcare and help people. I just always understood this stuff. Especially when I learned about DNA and RNA, I thought that was the coolest thing, that like these small molecules can determine, what we look like and also our health in the future and whether or not we have some sort of disease in the future. Um, so I attended university to get my bachelor's with the intention of either becoming a physician's assistant or a medical doctor. And then going through kind of the motions of seeing what that might entail, I decided that that wasn't for me exactly, but I still wanted to be in healthcare. So I kind of was confused because I had this biology degree, but I didn't really know where I wanted to be, whether I wanted to be in a lab, I wanted to get a graduate degree, go into public health, epidemiology. I didn't know. So what I did in my education, take several courses that revolved around genetics, specifically, and then also I did like recreating the yeast chromosome project in one of my labs, and got hands-on experience with these with laboratory techniques as well. And yeah, kind of thought I was going to go immediately into a graduate program, but then I decided that that might not be the best decision because it is a big financial and academic commitment to a graduate program, especially if you don't know what you want to do specifically. So that's when I decided, okay, I'm going to graduate and then I'm going to apply for jobs and see what I enjoy doing. And Shortly after graduating, I started my first position as a laboratory technologist in a molecular diagnostics laboratory. This laboratory specifically performed a variety of molecular tests, primarily on samples from cancer patients for initial diagnosis, monitoring disease progression, and detecting early recurrence of disease. There I learned how to extract DNA and RNA from a variety of sample types. I used capillary electrophoresis machines to run PCR for bone marrow engraftment, IgH, TCR, MSI, molar pregnancy testing. I also performed pyrosequencing, Sanger sequencing, next generation, and RT-qPCR. I was also trained to analyze all of this data as well, which is kind of how I got a brief taste of variant science and what that was, because about 3 years after I started, our volume increased so much, and technologists, more senior technologists, weren't able to analyze the variants and actually go through the data for the somatic variants and everything. So we actually started to hire variant scientists at that point. And then also during this time at this position, I got certified from ASCP in molecular biology. And then after 3.5 years working there, I decided to pursue a master's degree in biotechnology, since I thought, this is what I do enjoy doing. It's very interesting. I just enjoy working with my hands and then also doing science as well. And so I was doing the master's degree part-time while working full-time. And then in my kind of mid-career, I started a position as a molecular biologist in a microbiology lab. And while in that laboratory, I primarily ran routine COVID-19 testing. Since it was during the height of the pandemic, I also performed Sanger sequencing to identify bacterial species that had inconclusive testing results by other methodologies. So whether they were on a plate and you really couldn't tell by microscope, or staining processes what kind of bacteria it was. I also performed Sanger next generation sequencing testing to identify mutations in COVID-19 virus as the virus mutated into different variants like Delta, Omicron, all those ones that came out. And then I also did some testing and development for other microorganisms for the molecular microbiology lab. And I participated in some research project efforts. One of them was a digital PCR detection for COVID-19 mutation types. And then also helped one of our fellows with a rare bacteria species that had a mutation that caused it to not be toxigenic, which was interesting as well. So how I kind of came into variant science is I have a former colleague of mine who started as a variant scientist at Quest Diagnostics. And, whenever we'd catch up, she would I would ask her, how's the new job going? All this stuff. She would tell me about it. And it sounded very interesting, the position. And I was starting to be a little curious about maybe stepping away from my position, because it was just between the pandemic, being outside in everyday life, and then also doing it professionally every day, dealing with it. It just was draining, because it's like, okay, I'm just obsessed with the pandemic at this point, with all the work I'm doing. So I started to kind of look at jobs out of curiosity, and I saw that there was a variant science position available at Quest. And I reached out to my colleague to confirm that, it was her department, that they were looking for somebody, and she confirmed it. So that's when I decided to apply for the position. And it required 3 rounds of interviews. The first step was just a general chat with the manager, just to kind of see, what my experience is like. Also, compensation is also a conversation that was talked about initially, as in, just experience and why you're kind of interested in this position. And then a meeting with several of the other managers to see if, they agree that I am a good candidate potentially, or you're a good candidate. And then I presented an attempt at variant scoring to the managers and some of my potential colleagues, which are now my colleagues now. And the presentation portion is typically just to see if you have some basic understanding of evaluating variance and to give people an idea of how you can critically think and if you can kind of work through and logic the situation with the variant. I was offered, said position and have primarily been evaluating copy number variants and evaluating gene disease associations. And again, depending on the needs of the company, some job functions also include working on process improvement projects, aiding in testing new databases, and data sharing projects. So quite different, I think, than Yasmin, because I don't really have as much hands-on communication with laboratory personnel. Variant science, at least here, is a little bit more off to the side from the laboratory. And if there are conversations with the laboratory, that tends to be like a higher level conversation, not so much just like a regular variant scientist conversation. Yeah, and I guess, I've been doing this job for almost 4 years now, and I guess some advice for people who are interested in the field and interested in doing this. No one expects you to know it all, especially when you have no previous experience in this area. It's just really about showing that, you can rationale things properly and delegate your time correctly and are motivated to learn. That's, I think, the biggest thing that people really look for. Also make connections with other variant scientists or other scientists in general. You never know who's going to be in what field. Like I said, I was kind of a person that, luck of the draw that I had a colleague that I knew well. She knew, like, we both, had a lot of similar experience. So I didn't feel as intimidated to apply to this job with having no experience because I knew, oh, okay, well, we both worked in a lab together. We both have our master's degrees, or, I was still working on mine at the time, but like, in the process of it. So I was like, we have pretty much the same background. Like, yeah, I should apply to this. Um, so yeah, definitely always like keep your colleagues close, and always talk to them about what they're doing and what they're up to because you never know. And then also highlight your knowledge and how that translates into variant science. I think that is also a big thing because again, if you don't know everything, that's okay. But showing you can logic through problems and solve problems and use resources correctly, that is a major skill to have. And I can't reiterate that enough because I've definitely professionally run into people that are very confused on being resourceful sometimes. And so it's, extremely, extremely helpful to show those skills and kind of, even if you're in a laboratory role or if you're in an academic role or something like that, I think some of those skills can translate over and just making sure you're highlighting them when you're applying to these jobs and interviewing for these positions as well. And then I guess just general advice when you get into variant science, don't be afraid to ask your colleagues for help. Sometimes the content can be difficult to understand, and it only makes you a better scientist by learning from others and not suffering in silence. Or worse, completely misunderstanding the data and getting the wrong classification conclusion, which is not great. So you just don't be afraid to ask people, because again, we're all learning and not everything is always clear cut. And there's always conflicting data out there too. Sometimes, like, you'll read papers from 10 years ago that say one thing, and then this new paper will come up and say everything that was in that paper 10 years ago is not true. And you're kind of confused and you're like, okay, what should I believe? What is the good data and what is not? Um, so yeah, always don't be afraid to ask people for help. And then also being adaptable. Again, depending on the institution or the company you work for, there may be shifting needs. Specifically, like I know at Quest here, we are pretty, people can do different things, not just one role. And I know there are other companies that have people doing more so one role. So again, it depends on the place. But I think overall, just being adaptable to any problems that come up is always a positive attribute. Um, in this field. And then just when you think you understand genetics, you will probably evaluate a variant that makes you think, do I even understand it? Because I've definitely come into things where I'm like, okay, everything is saying that haploinsufficiency should exist for this gene, but then it doesn't. So what am I to do here? So again, genetics is such an evolving area, and our understanding is not always clear cut. So like, don't feel bad if you don't understand something. It doesn't make you a bad scientist. It will only make you a bad scientist if you don't actually ask yourself the questions and try to figure out what's going on. And then be willing to collaborate with different groups within and outside of your company or institution. I think, again, a lot of us go into this with the goal of helping to give people an answer to their disease, and being siloed off and not sharing information and restricting people's understanding is not going to help the greater humanity when it comes to like understanding disease and understanding these genes. So definitely be willing to collaborate with different groups, and either within your group, within your institution, or outside. But I think there's so many opportunities. Like, this is an existing opportunity now. There's ClinGen, there's a bunch of different orgs that you can get involved in that, you know, allow those collaborations to occur. And yeah, and that's it.
Thanks so much, Rachel. I think so many of us can empathize or agree with your assessments of learning something new every day.
Mhm.
And I also just want to reiterate that point about keeping your network open. And that's kind of one of the goals of this session itself, is to increase everyone's network. So thank you so much for bringing that up. Our next presenter will be Trisha, and she'll be talking to us today about her experience. Go ahead, Trisha, when you're ready.
Hi, everyone. Thank you for having me. My name is Trisha Hebron. To start out with, I just needed to show this conflict of interest slide. I'm an employee of LabCorp. And just a little bit about me to start out with. I'm based in the Los Angeles area. I'm originally from Northern California. And as I just mentioned, I'm currently a variant scientist at LabCorp, formerly Invitae. I'm a genetic counselor by training, and I've moved around to various settings, such as diagnostic labs. I've been in the academic and hospital settings as well, which I will be highlighting for you. So to start out with, I did my undergraduate studies at the University of California, Davis. And to be quite honest with you, I chose my genetics major very blindly. I was very naive, just excited to be moving out to college. My best friend at that time chose genetics. Genentech and Roche were big employers in my area, and I just said, why not? Let's choose genetics. I could always change my mind. And so I didn't really know what to do with my major after graduation. I kind of weeded out med school. I knew I didn't want to do research. I didn't want to do wet lab work, but I really loved my major. I really ended up loving genetics. And so at that time, I was doing a part-time job, doing some pipetting as a research assistant at a DNA sequencing lab on campus. And my manager at that time, had mentioned genetic counseling. I had heard about genetic counseling in some classes here and there, but never really thought much of it. But my manager at that time said, hey, I think you'd be great at it. You should check it out. And so I shadowed some pediatric genetic counselors at the UC Davis Mind Institute and ended up falling in love with it. I applied for genetic counseling master's programs and fortunately got in. I attended the CSU Stanislaus program, which no longer exists. It's now the UCSF program. And I graduated in 2014. I landed a job soon after as a clinical pediatric genetic counselor at UCSF and really thought that this would be my forever job. I thought UCSF is a great place, and just thought I would be there forever. Um, Worked in a general genetics clinic. That was kind of my home base. I saw families affected by a variety of different genetic conditions, pretty much every condition except for hereditary cancer and prenatal. I was also very much involved in specialty clinics like the hearing loss clinic. We had a rasopathies clinic. And also, we had a new whole exome sequencing clinic at that time. And this was around the time when whole exome sequencing was just sort of new to the clinical setting. I really enjoy digging into these reports that I received for my patients. I love digging into why a variant was classified in a certain way, and it really piqued my interest in applying for industry roles. And so my what I thought was my forever job all of a sudden didn't seem like forever to me anymore. I was also getting tired of commuting every day, and so a hybrid role really became more and more attractive to me. And this is how I ended up at Personalis. So I landed a job at Personalis, which is a precision oncology company. And so at that time, Personalis was doing whole exome sequencing, germline testing. And within a few months of me getting hired, they actually decided to switch gears and completely just do somatic tumor testing, which is similar to what Yasmin is doing. Um, this was very new to me, not something that we had learned at all in genetic counseling school. Genetic counselors are primarily trained in inherited conditions when I was in school. And so somatic testing was not something that was really on my radar initially. But, I leaned into it, loved doing it. At that time, this was something that was really unique as a genetic counselor, and I love being able to say that I was doing something very different. And so I built my expertise in this and did this for over 7 years. I didn't have a variant scientist title at Personalis. I was actually a senior genomics counselor, but I certainly, the majority of my job was performing variant interpretation and clinical report writing as my main roles. And it was something that was in the survey that Alexa had showed earlier too. I wore multiple hats. I was involved in things like assay validation, helping marketing launch our new tests, refining our processes. But unfortunately, after about 7 years, I ended up becoming part of a layoff, which is unfortunately something that, we all take into consideration when working in industry. It took me, I would say, over 5 months of job searching in a brutal post-COVID market. Um, Eventually landed a job at Cedars-Sinai's molecular pathology lab. Um, and they were hiring their very first variant scientist, which was very exciting to me. However, this required me to be on site every day. It required me and my husband, our two dogs, to pack up our lives in Northern California and move down to Los Angeles. Um, but it was a very exciting role. I helped them, standardize their variant interpretation and reporting processes. I was heavily involved in educating molecular pathology fellows about variant interpretation. Um, However, I had a life change. About 6 months ago, I had my daughter and really needed to be remote. So fortunately, was able to land my current job at LabCorp Genetics, which was formerly Invitae. And I'm currently a variant scientist on their clinical genomics operations and support team. Something that is great about this line of work, right? There's lots of opportunities for remote work if this is something that fits into your lifestyle. Um, and so I'm switching back to doing germline testing after over 7 years. After I'm done with my training period, I will be working on doing more variant interpretation for various in vitro test offerings the majority of my work will actually be helping to help refine our processes and answer scientific inquiries. Um, and so, I really enjoy what I do indirectly, helping patients with, their diagnostic odysseys. I get to just kind of what Yasmin and Rachel had mentioned, I get to nerd out and dig into different variants and different genetic conditions. Um, get to be involved in improving workflows and processes, which is something that I really enjoy. And again, the opportunity to work remotely and have a job that fits my lifestyle is really invaluable to me. No long commutes and just more time with my family. And just to tack on to what Alexa said earlier, if I can just give some advice to utilize your network, especially this one. And really just, if this is something that you're interested in doing, seek shadowing opportunities at various settings because it really does look different in different settings. Um, and LinkedIn is such a powerful tool if you just want to reach out to variant scientists and ask if you could shadow. And if you can't, I'm sure that they could direct you to somebody who might be able to help. So thank you for listening, and I'm really excited to be part of this Variant Scientist Network and hope that you all will take advantage of all the exciting things that we have in store.
That's so great, Trisha. I appreciate that so much. It sounds very much like all of us had very similar experiences in getting into variant science. So I'm going to open it up for questions now from the audience. Please feel free to raise your hand, virtual hand in Teams, or type it into the chat or type it into Q&A for questions. And I'll get it started because so many of us sound very much like we did transition from one field to variant science. Does anyone want to take a question about how you would encourage someone who's in that position transitioning from one field into variant science? What are the kinds of things they should do? There was a question in the chat about ClinGen, and that's something that I definitely endorse. Do you all have any recommendations? Any of Trisha, Rachel, Yasmin, do you want to get it started?
I mean,
I think, oh, please go ahead.
Go ahead.
Go ahead.
No, go.
Okay, so I was going to just point out that the resources that are available on the Variant Scientist Network website. So I'm not sure if this particular one is there, but there's some great YouTube videos, and there's also something called Future Learn, where if you want to explore and learn more and understand the steps of how variant interpretation works, there's some great videos out there. That was something that I did once I was already in my position, which I found helpful. But if I had known that they existed before, I think, and I'd watched them before, they would have really helped me to kind of narrow down what it really was about at the time. And then I'm just going to reiterate, right, is just talk to your colleagues and fellow scientists. Ask them whatever question you need to ask them. I think we're all pretty open people. We like sharing, we like helping. So, don't be shy. And we're all very giving of our time, I think, with obviously our limitations, but and this is the goal of the Variant Scientist Network, is to try and help everyone here in your own journey. So I think that answers as much as I can right now. I have other ideas, but I'll stop now.
I mean,
Yeah, I think. Oh. No, go ahead, Rachel. Sorry.
Oh, okay. Coming from like a wet bench experience side of things, I think it is very important to ask management questions as well to get involved in things. Because sometimes people don't know what you want, especially if you do have variant science like roles at your current position or your current place of work. I think it could be easy for a manager to say, okay, yeah, you could sit down with so and so and shadow them, or even just bringing it up to management, because I know some places they may only have one person doing it, and then say, hey, like I might be interested in this, like can I maybe get some experience like helping them out at some level to get your hands wet. I mean, I feel like that's kind of how I got a mild introduction. Was I was doing data analysis for NGS, and sometimes they would ask me like, hey, can you just like check Cosmic or check this database to like see this data to see if it's something relevant or not. So that's kind of how I did it, was like more volunteering and asking the questions. But again, like Yasmin said, the resources too in ClinGen, I didn't know they existed before I started either. So I capitalized on them once I started my role.
That's great. Thank you all for that answer. I think we'll transition now maybe to in-person questions. Maria, do you want to go ahead and go first?
Sure. Thanks for organizing this, Alexa. I have two questions. The first one is for you. So as you were doing the survey, preparing the publication, I don't know whether you addressed this. What I'm experiencing now is I don't see much career progression or upward trajectory that's very clear or defined for variant scientists. Did you address that, or what ideas or suggestions do institutions have for people like us? Because most of our duties seem to be the same year to year. That when you go in to even argue for promotion, they're kind of like, oh, you do the same thing year to year. It's kind of hard to define it, what that next step looks like. So maybe if you could address that, whether you found any of that in your study. And then the second question is for Tricia. And maybe Rachel can also answer this. Maybe you could give us some pros and cons, or describe the differences or similarities in your experiences as you transition from an academic or hospital lab versus working in a diagnostic lab in an industry setting.
Those are great questions, Maria. So I'll answer the first one, and then I'll touch a little bit on the second one as well. So the first question was about career progression or career ladder for variant scientists. And in many places, that just does not exist, right? At least in our experience. We did not ask specific questions about that, but we are meeting this afternoon to start our follow-up survey, and that definitely will be one of the follow-up surveys focuses. I do have some slides that I'll see if I can share with you that we did touch on the very first variant science ACMG session. We touched on possibilities for career progression. Different organizations have set up some sorts of career progressions in an analyst to a scientist to a scientist to a senior scientist kind of career progression. And I think that that's something that organizations should look into to keep people like us that are highly educated and highly experienced involved and at the same position. Because often when we think about these kinds of things, the first recommendation that we're given is to go do a fellowship to become a medical director. But for me specifically, that's not something I'm interested in because I enjoy what I do so much. I don't want to necessarily change what I do, but I would like to be recognized for the decade plus amount of experience that I have. So that will definitely be the focus of the next survey. So keep your eye out for that. And then Tricia and Rachel, if you want to touch on Maria's second question.
I was actually going to suggest to Maria's first question about, at least here at Quest, we have created a career ladder that's very clear with like what you need to achieve in order to become a genomic variant scientist too. And we kind of recently changed some things around as well, where now we have technical leads, so people that are really specialized in a specific area. And then we have managers and senior level managers, and then we have our director. So we've created a ladder structure that's very clear on what you need to do in order to get from a regular genomic variant scientist to a genomics variant scientist to a technical lead. And again, all of these things need to be justified within the corporation, and that's kind of, I think, difficulty with some industry stuff. Unfortunately, there's sometimes a lot of red tape. But then again, I guess on the academic side of things, it also sometimes is difficult to vouch for new position creation. Like, again, I worked in a hospital setting, and it was there was one time we had a roundtable and we were like, can we have a molecular technologist level 2 position that exists here? And they're like, no, we'll just pay you more if that we want to. We don't want to necessarily give you another title, or create this structure to give somebody another title. So it can be challenging, but I guess, yeah, you got to work within the organizational structure. But yeah, I think it is helpful to have clear guidelines on what the expectations are.
Okay, and then I think her second question was transitioning from one kind of career to another. And I'm going to approach it from the other direction where I started in a corporate setting and transitioned to a more academic setting. There are differences. I'm not sure, I don't remember exactly what your question was in regards to, but there are differences. And it's just kind of what Rachel, I think, touched on in being flexible and being willing to adapt to different situations is very helpful.
And I think one thing with industry, I think, is that there is a little bit of a push for faster turnaround times. That can be a thing that exists, as well as we sometimes have difficulty with getting good phenotypic information for the patients. So when you're in a hospital setting, you have the luxury of being able to go into Epic and check the patient records and see what's going on, and see that they've gotten a X-ray or something like that to confirm they have this, abnormal skeletal structure or something like that. Like, we don't typically get that stuff. So definitely is more challenging, I think, in that aspect, when it comes to clinical care. Because, like, of course, we want to give the best care, but unfortunately, we're kind of bound to turnaround times and limited patient information.
Yeah, and I think for me, my roles at these different settings were very different, particularly, when I was in the academic hospital setting versus in the diagnostic lab. When I was in the hospital setting, I was a clinical genetic counselor, and I loved seeing patients, but I really found myself being more and more interested in the diagnostic laboratory side of things. At that time, we had to be fully in person. When I was working in clinic, I believe now a lot of genetic counselors in the clinical setting can also see patients remotely, but at that time I had to be fully in person. Industry allowed for me to be able to work remotely, which was really important for me at that time. Um, One thing about just tacking on to joining industry and having transferable skills, I didn't have any variant science experience specifically when I joined industry, but what I really liked about being a genetic counselor jumping into industry is that I was really able to be an expert in pediatric genetics and really felt like I added value to the team. Um, One other thing, we were talking about flexibility being really important. I joined Personalis to do germline testing, and within a few months, they switched to doing somatic testing. And I think, if you're going to join a startup, an early startup, such as the one that I joined, you have to be really open to that, and also having to wear multiple hats. I actually really enjoyed working with various teams. I was being pulled into bioinformatics meetings, marketing meetings, just a variety of different teams. And if that's something that you really enjoy, then that might be something for you. But some folks really like just doing one thing, just really doing casework. And then finally, a con of, I would say, working in industry, coming from somebody who has been part of a layoff, thinking about being in a hospital setting, being more stable, um, for the most part, I think, than, for example, joining a startup lab, which might be a little bit riskier, is something to think about.
We have several hands raised. I think we may be able to get to at least one of them. So, Raya, go ahead.
Yeah. Hi, my name is Raya. I'm currently getting my master's in human genetics and genomic data analytics at Keck Graduate. I guess my question is, I'll be graduating in the spring. I have this, since I've been in my program, this background in how to interpret variants, SNVs, CNVs. I've done the NGS sequencing. I'm just trying to figure out now just how to structure and like highlight these things on like a CV or a resume. So if any of you had like any tips for like how to proctor myself, knowing that this is academic experience per se, rather than like actual industry experience.
I love that question. As someone who's been hired or has hired in the past, I would like to say that I think having a resume rather than maybe a CV is good. Personally, I like to see resumes. And I think that although your experience is academic, having those things highlighted, the types of tools and resources that variant scientists use on a daily basis, even if it is just on an academic basis in classes, that's still experience that you have that other applicants might not have. So I would list those things specifically that you have experience with those things. And maybe if you could self-assess your proficiency in those types of areas, or maybe give examples of projects or those sorts of things that you can highlight to show your experience in those resources, I think would be good. Does anyone else have any comments on that?
I think this is probably jumping like one step ahead, but in an interview, I think, I really love when I'm interviewing somebody and they can provide me with a specific example of an interesting or a challenging case or a variant that they've worked on. If you could walk us through a variant or a case, highlight any issues that you've come across and how you worked through it, I think that is something that's very powerful to me.
I think again, it's kind of back to what you said. It's just like that, having that innate resourcefulness and wanting to look up the information and find the information.
We have only
Thank
3 minutes left in the time allotted, and it looks like there are so many more questions that people have. While we ask the last person who has their hands up, if while you ask this question, I'm just going to put on the last slide. Be on the lookout for a post-event survey that we will be sending out to everyone, and save the date for our first in-person networking event that will be during the ACMG meeting in Baltimore. Okay, so Dipti, if you want to ask your last question, we'll have your question and then we'll wrap up.
Okay, thank you so much. So I just posted a new message also. Uh, I'm just curious about these AI interpretation tools that I'm seeing every day coming on LinkedIn or other websites, and there are so many platforms available now. Is it going to be a challenge for a role of a variant scientist, starting at a, let's say, at the beginning of their career?
Personally, I kind of answered a
Yeah.
AI question that was earlier on. I really think that someone beginning their career in variant science now has an awesome opportunity to incorporate all of the AI tools that are available at the beginning. So I think honestly, in some ways, you might have an opportunity to be the first adopter. I do think that with AI, knowing which AI tools hallucinate and which don't, which ones are
Mhm.
useful and which ones aren't, those sorts of things are really critical. And someone entering the field might not have the knowledge to know which ones are reliable and which ones aren't. But if you
Mhm.
can do your own independent work on that and determine for yourself which ones hallucinate, which ones are reliable, which ones are useful, I think that you have an awesome opportunity to be an early adopter of that and incorporate it into your work and make you more streamlined and more I don't know, but more fast paced maybe than some of us old folks that have a hard time integrating AI. That's my perspective. I think that AI will revolutionize variant science in that it will make us more effective and streamlined.
Mhm.
I don't think it will ever be able to effectively analyze literature critically to be able to say that what the report says it shows actually is what it shows. And then the other thing that I think it still struggles with is to be able to incorporate various different types of information into a coherent structure, specifically in electronic medical records. I still haven't seen one that does a good job at doing that and incorporating that into reports. In the last minute, do any of our panelists have anything to add to what I said?
I completely agree with you, Alexa. And then also to add, it's just not quite there yet when it comes to the analysis of these papers. Um, I funny thing that happened is we got a genetic counselor because I primarily work on CMVs, and they used one of these research AI tools, and they pulled up a bunch of random papers. Like one was on tetrasomy of this region. There was one that was like a 10 megabase still or duplication that had nothing to do with the region. So like they're not good at parsing out the information right now. Unfortunately, you still need that human component, especially when it comes to CMVs. They are definitely not there at all. But yeah, like I mean, it was funny. I like responded to the genetic counselor and I was like, yeah, these papers don't cover the region at all that we're talking about. So like there's really no information out there on this specific, I think it was like 5 or 6 genes and it was like maybe 2 megabases or something like that. But yeah, it was looking for like anything that just had this cytoband and duplication, and that was it. And it was just threw you all the trash. So it's still not there, especially for
Yeah.
CMVs, I will say.
Okay. Just it's I'm just I just want to learn how what we can do about CNVs and if we can. So I work with a company who actually works on the AI side of it. We are actually trying to find out a solution for CNV analysis. So whenever there is a future meeting, maybe we can, if there is a possibility to discuss what exactly we expect as a variant scientist to, how to make these things visible, which will actually reduce the workload for everybody. So
Great.
yeah, thank you
Thank
so
you.
much for the insights. Yeah.
Thank you. All right, there's a link in the chat if you're interested in submitting for an ACMG Lightning Talk that we'll be having at our first networking event. Please keep your eye out in your inboxes for our next Variant Scientist Network event, which will be next month. It will be an educational event on RNA. And thank you so much for everyone attending. We appreciate the participation. Look forward to seeing you in the future.
Thank you.
Thank you, everybody. Thank you.